各位朋友，大家晚上好！欢迎大家来到新中科技论坛。新中科技论坛是由新加坡、中国科学技术交流促进协会主办的。新中科促会致力于在新加坡和中国之间为科技工作者、科研人员、专业人士等搭建知识与科技交流互动的平台。我们邀请全球知名的专家与学者，在这里为大家进行不同学术、不同专业领域的知识和经验的分享，也积极推动两国科技与产业融合发展。在此，我们欢迎各位朋友加入我们的协会，让我们共同为新加坡和中国的合作发展做出努力。如果想进一步了解我们协会的情况，请登录我们的网站 scast.org.sg。可以了。20世纪至今，人类社会已经发生了5次科技革命，宏观至天体运行、星系演化，微观小至基因编辑、粒子结构和量子调控。与基因科学有关的科技与应用，无疑对人类社会发展产生了深远的影响。新冠疫情的爆发，让人们对基因科学的发展走向也更为关注。今天我们邀请马来西亚的基因科学领域的专家 Dr. Cole， 请他为我们介绍一下现代基因技术在人体健康管理、疾病防范等方面的作用。Good evening, ladies and gentlemen. Welcome to Singapore China Science and Technology Forum. It gives me great pleasure to introduce Dr. Cole from Gene Express Private Limited, whom is an expert in genetic science, research, and application. Let us all welcome Dr. Cole. Dr. Cole, the floor is yours.
Yeah, thank you very much, Chairperson, for your introduction. So sorry, I scared of one stop.
It's okay.
Okay. Well, good evening, ladies and gentlemen. Sorry, I got the wrong file up. Yeah. So this evening, it's my pleasure to share with you on genetic testing. I will give you an overview. So the topics that I will be covering will be on some basic concepts, and then only we'll talk about genomic variants, and followed by the genetic testing methods, various genetic tests which are available, and also some ELSI, which is the ethics, legal, social implications of genetic testing. Okay, now our body, we are humans, and we are what we call a multicellular organism. In other words, we have lots of cells, trillions of cells. Okay, so if you look into our body, you'll see lots of cells. And then you can see in the middle here, there's some dark spot there. That's the nucleus. And the nucleus contains the genetic material. What happened to mine?
What's wrong?
What's wrong with my screen?
黄教授，你把您的屏幕要关掉。
黄教授现在在 share， 他把share screen 要关掉，他那个 overlap，
对，你要关掉，不然的话，因为你是 host， 因为黄教授是 host， 他可以把 Dr. Cole 的给Dr. Cole， 您可以 you can share your screen again, Dr. Cole.
Yeah, okay.
Sorry about it.
No, it's okay. Yeah.
It was an accident, I think.
Yeah, it happens all the time.
Sorry.
It's okay. Okay, I can see my screen now, right?
Yes.
Yeah.
Yeah.
So as I said, if we look into our body, we have lots of cells all over the body. And you can see each cell here in the middle, there's a dark spot, and that's the nucleus. Within the nucleus, we have chromosomes. Chromosomes either in a form which can be drawn like this, linear, or in a form, it's like an X here, okay? There's divided chromosome. Now, chromosome is it carries our genetic material, and it carries genes along the length. And if you look into it, it's the DNA. So that's a level. So DNA is actually the genetic material. When we look at a particular cell, now it's being broken up here, bigger. So nucleus has a cell chromosome here. And this part here is called the cytoplasm. In the cytoplasm, we have another small structure called mitochondrion, which also carries DNA, but the DNA is circular, mitochondrial DNA. In other words, in a human cell, we have two types of DNA. One type from mitochondria, one from the nucleus. But the main bulk, most of the DNA is in the nucleus. So if we take the DNA here and the mitochondria, that forms the genome. Genome means the complete set of genetic material in a cell from the nucleus and the mitochondria. So if we expand, look into bigger one, the DNA here, as you'll see later, it has DNA, and DNA is made up of these chemicals. So before I go further, I also want to explain our cells. In our body, we have two types of cell. We have the diploid cell called the somatic cell. Soma means our body. And in each cell, inside here, if you can measure them, there are 46 chromosomes. It's actually 23 pairs. There are two sets of nuclear genome. On the other hand, every human also has what they call the haploid cell. These are our sex cells. In the female will be in the eggs, in the male will be sperm cells. The sex cells has half the number, 23 chromosomes. So they call one set of the genome, nuclear genome. So diploid cell has two sets of the nuclear genomes because 23 pairs. And then of course we have the mitochondrial genome here, mitochondria. So that is our genome, okay? Now, DNA is our genetic material. DNA stands for deoxyribonucleic acid, but we normally call it DNA. So as you see there, DNA, it is a twisted structure. And if you look at it carefully, if you pull it and stretch it out, it has two strands, and one strand of black and one strand blue here. So there are two strands, two linear strands. So this is called a double-stranded DNA. And if you look carefully, it is made up of 4 different building blocks, A, C, G, and T. So these are the 4 different bricks that build up DNA. And this is the language of life. Very simple, although DNA is very complex. So there are 2 linear DNA strands, and they are arranged in such a way that we say that they are base pairing. It's not random. They base pair with each other. What does that mean? You can see that wherever there's an A, you have a T. A, T, A, T. So A will always pair with T, and G will always pair with C, GC. So it's called base pairing. And this is called complementary base pairing, complementary to each other, okay? And we call this the complementary linear base sequences. So this is what they call a base sequence, shown here or shown here. Now, the double-stranded DNA here, linear, can be twisted. You twist them and they form what they call the helix, the double helix, the famous double helix. Now, this base sequence, it looks very simple, but it's very, very important. It's also called the nucleotide sequence because it is genetic information. This one has meaning to a cell. It's actually a gene. A gene means this way, one stretch of nucleotide, okay? So, for example, you have thalassemia genes or you have ABO blood, the genes that control our blood group. So now we have the technology to determine the entire sequence from our genome. So that is called DNA sequencing. DNA sequencing means to determine the entire DNA sequence, the order within a DNA. Okay, it can be one piece of DNA or the entire genome. So we now know that the genome in a human is made up of 3 billion base pair. One genome is 3 billion, and that is in the haploid cell. In the diploid cell, we have 6 billion because multiplied by 2. Okay, a lot of thing, but now can be done. So the information here in DNA, of course, must be used. So the genetic information stored in DNA must be expressed. When you express means can be used. In other words, used to carry out cell functions and metabolism. Cell function and metabolism are carried out by different types of proteins. There are different kinds of proteins in our body. In our body, we can classify proteins into structural. They give us structure like tendons, cartilage, hair, our nail. When you look at each other, we're looking at our proteins. And look at the contractile muscles. Muscles we can contract. And then you have transport protein like hemoglobin, myoglobin that transport oxygen. And then nutrients, some serve as food, hormones like insulin, enzymes. And then very important for protection. These are the antibodies, especially during COVID period, our immunity. All these are proteins, different types of proteins that carry out a function, okay? And of course, among them, enzymes are very important because for us to be alive, we must have metabolism. Metabolism means vital life processes that sustain life. Metabolism means you can synthesize, you can build up bigger molecules, or you can break down. So in our body, two things are happening all the time: building up, breaking down. So digestion, for example. So, and all these are carried out by metabolic pathways. There are pathways. And for example, these are the complicated pathways. So all these pathways are happening right now in our cells. It's very complicated. Just to show you, not in the detail. So, there is a lot of pathways. When you talk about pathway, we have one compound converted to another compound to another compound. So it's a substrate to a product. And every step is carried out by an enzyme. So that's why enzyme is very important. So in our body, we have more than 75,000 different enzymes to carry out all the functions. And enzymes sustain our life, healthy life. If one or two enzymes, especially the important enzymes, are not working in our body, then there's a problem. You have what they call genetic disorder. Okay, so all these proteins, all the proteins here, they are encoded by genes. It's a DNA. All the DNA here, they carry the message to synthesize them. Okay, so that is the link between DNA and proteins. So how does DNA make protein? Now, a certain sequence in DNA is used to make a particular protein, not directly. It goes through an RNA intermediate. So we say DNA makes RNA, RNA makes protein, DNA. So these are three very important biomolecules: DNA, RNA, protein. They are big molecules, and each of them is made up of smaller molecules, monomer. Like the nucleotide DNA, we have shown you 4 types: A, C, G, T. RNA has A, C, G, U, no T but U. And amino protein is made up of 20 different amino acids. Okay. So this is a process. If you can remember this, you know a lot about molecular biology already. So DNA makes RNA, makes protein or polypeptide chain. The process is DNA that makes RNA here is called transcription. And then once you got RNA, RNA will make protein translation, like translating a language from Chinese to Russian or to English. So this is how you get your protein. So you can see here, when you look at a polypeptide or protein, the types, these are amino acids. The number, how many there, the type there, whether it's glycine, whether it's alanine, and the sequence. This is not random. It's all determined by the message, by the DNA sequence. The sequence in the DNA here determines the sequence, and it executed through RNA. So DNA sequence determines the sequence of RNA, and this determines the sequence. That's how the link. And that's why nucleotides is very, very important. You cannot simply change them. Okay, so you know the basic. And now gene expression is very important. When people talk about gene expression, it means from DNA to RNA. When this step, this is talking about gene expression means transcription step. And this step can be switched on or switched off. It's not on all the time. It can be like there are switches. They can switch it on, switch it off. And once you get the RNA, some RNA or most of the RNA will become templates. Template means like a mold that can be used to make protein for translation, but not 100%. Some RNA will remain as non-template. It remains as RNA. In the cells, there are RNA for other function. So gene expression does not always lead to protein. This you must understand. So there are many genes. The take-home message is there are many genes in our cells, in our body. All the cells in your body, they are identical. However, they are not expressed at the same time. So they are expressed according to time. For example, when you are a baby, before you are born in a mother's body, fetus, certain genes are expressed. When we are born as a baby, certain genes are expressed. When you are young, when you're old, at different stages, different genes are expressed. That's called time. Space. Space means location. The cells know. In your brain, they know that they must behave like a brain cell. In your liver, like a liver cell. In your heart cell. Certain genes will be expressed. Okay? And environmental condition. So sometimes the environment affects expression. So we call the spatial-temporal gene expression. So regulation of gene expression is very, very important. You cannot afford to have genes simply expressed all over the place, and it can be controlled. Okay? In other words, transcription can be switched on, switched off. So in other words, if you are parents, you must be very grateful if for proper gene expression to have a proper child or a child with all born normal and healthy. For example, you can have a child born like this. Imagine a child born with so many toes. Gene expression has gone wrong. So you can see here. Or you can have a child with an extra arm. Because here, certain genes have gone wrong. They forgot. They should only have 2 pair of arms, but now they got extra. And then worse, you have in a body, you have another body. So these genes express. So, you know, gene expression goes wrong. And there's a lot of problem. You don't want your brain cell suddenly have an arm or horn coming out, you know, things like that. So gene expression is very, very important. So with this as the basis, I now go to the proper about my talk today about genetic testing. What do you mean by testing? But before that, let me say about genetics and genomics. Genetics is a science of inheritance. So how genes are transmitted from one generation to another. Talk about genes. Genomics now, because of the advances in technology, we can study many, many genes at the same time. We can study the entire genome. So it is the science of structure, function, evolution, mapping, editing of genomes. So you are talking at the genome, you include the interaction. So it's very complicated now, genomics. So now, for example, in the last 2 years, there are many terms in genomics. A lot of people are familiar with. They're familiar with because of what people watching TV program like forensic DNA, and then all these terms. Many of you are familiar, especially like variants, sequencing, genomes, PCR. It's related to the SARS and COVID. You go for your test and you go for this one. Okay, so most people are familiar with. So now let me talk about the term. It's very important to understand genomic variants. So it's talking about normal. Normal, you refer to, actually there's no such thing as a normal. We have a reference genome for comparison. So normal versus variation. Variation means change. So this variation actually comes from mutation. But nowadays people don't like the term mutation because people think of mutants something strange, like 怪物, you know? So people use variation. But variation actually comes from mutation. And then your variant is referred to mutant, but can be interchangeable. So when you talk about genomic variant, there are three levels of variant. One at the DNA level. At the DNA level, we have single nucleotide or base change. Individual, as you know, there are 4, ATCG. Either you call it a base or nucleotide. They can change. So for example, originally it's an A, and this A can change to C or G or T. So this is a mutation or called a variation. Or you can have insertion, one extra being added in, or deletion, one being taken out, one or more. So at the DNA level, at the bigger level, chromosome. Chromosome means large genetic change, including copy number variants. I'll explain to you later. So here it involves the entire chromosome or certain part of the chromosome change. So you have extra chromosome missing, addition or deletion. Or you can have the chromosome, certain parts of it rearranged. They are rearranged. So, so this one, we are looking at numerical changes in number or structural change at the structure of the DNA. So abnormality, anything that changed the genetic material, at the end is still DNA. Chromosome has DNA. It has changed. Or you can look at protein. Although protein is not DNA, not genetic material, protein, they are products. They are products of genetic material. So when you look at the protein, you have an idea. If there's an absence of a protein, important protein, you know the gene, something is wrong with the gene. So you can deduce. Okay? So this is a very important, this part is a very important diagram that shows the variance. So as I told you, at the largest level, you have a chromosomal variant. At this chromosome, you can have duplication, certain parts duplicated, that means added in, additional parts added in, or deletion, certain part removed, inversion, certain part turn around, just like your English language sentence, you turn around the sentence, the meaning has changed. Translocation, that means one chromosome, they exchange. One part from here go to this chromosome, they can exchange, or they can go expansion, certain part expand. So all this can happen to a chromosome. That's called a structural. Numerical means extra chromosome. One extra chromosome, or here one chromosome less, the entire chromosome. And then at the lower level, you have copy number. We talk about copy number variants. There are certain gene sequences, they will expand. They will have additional sequence. So, and then at the lowest level, at the DNA level, then of course you have the nucleotide variant. So you can have the base change. One can change to these 3. So this is also called, it's called base substitutions. And it can be insertion, one can add additional, or you can delete. And if it involves substitution, it's also called SNP, single nucleotide polymorphism. That means variation in one particular nucleotide, many of them for that. So you can see here copy number, or copy number can also talk about copy number variant region. There's one region with copy number variations. Copy number variants involve repetitive DNA sequences. Certain sequences are repeated many times. Just like here, you can see here the simple one, trinucleotide repeats. There are trinucleotides. There are 3 nucleotides being repeated, GAT, repeated. There's a small number, but this one normally is a bigger chunk of DNA being repeated. Okay. Now, once we know this, then we can look at, once you understand what are the variants, now we look at genetic testing. Genetic testing, by definition, any test that examines DNA or one or more genes, you can examine RNA, you can examine chromosomes, or examine proteins. That it comes all under genetic testing. So let me first look at or let us now concentrate on chromosome, genetic testing of chromosomes. So we have different types of chromosomal genetic tests. So these are known as diagnostic. Diagnostic means can be used for medical treatment when you have diagnostic. That means a doctor will do a diagnostic. Anything test not for medical treatment is non-diagnostic. So chromosomal tests are normally meant for diagnostic. That means the results will help to for medical treatment. So they are referred to as diagnostic cytogenetics or cytogenomics, or molecular cytogenetics, cytogenomics. So there are 3 types here. The first type is karyotype analysis. This is historically earliest type, traditional cytogenetic test. Cyto means cell. You look at genetic at the cell level. This one involves tissue culture. You culture the cell, and then you encourage the cell to go into mitosis. Mitosis is cell division. And then you need to collect chromosome at the metaphase stage. Metaphase is a particular stage where the chromosomes are very thick. You get chromosomes very thick and easy to study. And then you can do chromosome banding and use microscope. I will show you afterwards. The other technique, more molecular, is FISH, or fluorescent in situ hybridization methodology. This one uses molecular technology, and you can analyze interphase cells which is not dividing. That means cells are not dividing. You can analyze them very quickly. This one will make it is a very fast method, increases the speed and diagnostic resolution of clinical samples. And the other method is chromosomal microarray. I'll show you afterwards. So there are three major methods for chromosomal DNA tests. And they will allow us to evaluate numerical, that means changes in number, or structural chromosomes. And then we can define what kind of rearrangement, especially the structure. So here we have two examples of karyotype. So we get a metaphase chromosome. These are metaphase chromosomes, very short, very thick. And these chromosomes have been treated with enzymes and stains. And you can see they are bent. So it's called chromosome bending. The chromosomes, you can see bands. So that allows us to match them, to pair them. So somatic cells has 23 pairs, 23 pairs. This one is for male, X and Y chromosome, X and Y. For female, you have XX, okay? Now, this is the normal human. But when you look at this one, if you look at chromosome 21, you only a pair here, but this one has extra chromosome, so +1. So this is what they call trisomy. There are 3 chromosomes, 21, and that is a disease. The disease situation is called a Down syndrome. So you can use this, but this is a lot of work. You have to tissue culture the cells, you have to put them into mitosis and do microbending microscope. And then you do careful pairing, a lot of work. But from there, you can do what they call the aneuploidy. Aneuploidy here means whether got additional chromosome or loss of chromosome. So these are some common diseases, human diseases, that can be detected based on karyotype analysis. So normal human, male, 46 chromosome XY, 46 XX female, but 1 chromosome missing, only X, is called Turner syndrome, monosomy, 1X, or you can have XXY, double X and 1 extra Y. Klinefelter syndrome is a male. The moment you have Y chromosome, you are a male, even though you have 2X. This one you have XYY, double Ys, Jacob's male. And then you have male or female with extra chromosome 21. Chromosome 13 is called Patau syndrome. Extra chromosome 18, Edwards syndrome, or extra X meta female. So all these are referred to as genetic disorders that can be determined through chromosome studies. But now with FISH, it has also introduced the technique of chromosome painting. We can paint chromosomes into very colorful appearance. This is all based on dye, fluorescent dye being used laser beam to excite them. You can see this. So this is called diagnostic cytogenetics or clinical and molecular cytogenetics. So this is a normal male, X and Y, nothing. Okay. And then the good thing, as I told you, in FISH, you can use any cell. You can use a non-dividing cell. You don't need the very condensed chromosome. You don't need the metaphase chromosome. Like this is a normal chromosome cell. Inside the nucleus, normally is a dark. You can stain them. You can use special probes, DNA probes, to detect them. Like in this case, and paint the chromosomes. You can see 2 green dots. 2 green dots means there are 2 green chromosome 13, normal. But the 3 red dots refer to 3 chromosome 21. So when you see an extra dot, it's bad news. This is additional chromosome, Down syndrome. Or you can have a metaphase chromosome like this. You can see here, 1 dot, 1 dot, 1 dot. There are 3 yellow dots. Every dot will pick up chromosome 21, 21. So very quickly, we know it is also extra chromosome 21. So this is the advantage of FISH, very fast. We can detect them. So I've covered 2 methods, the microarray, the FISH method, and then the karyotype method. So the third method is microsomal microarray. This is microarray means you have a microscopic slice. On here, you have thousands of millions of dots here. Every dot, you have DNA sequences. And these DNA sequences, they're specially made. And when you take patient DNA and the control normal DNA, mix them and put on here, they will mix with them. And at the end, after treatment, you can see spots. So you can see these are spots. So microarrays, then the spots, because the DNA has different dye, fluorescent dye, the patient is green label, this is red label, you will see yellow spot, green spot, or red spot. Yellow spot means a mixture of the 2, hybrid. And from there, we can detect. For example, you can see this means extra DNA. Extra DNA, this means less DNA. When you see a peak down, just I think down, there's a deletion. There's a deletion. This is duplication, additional. Extra chromosome here, and not enough chromosome. So microarray refer to a microchip-based testing platform that allows high volume automated DNA analysis of many DNA at one time. That's the advantage of microarray. It's a molecular technique. So the chips can be, they use the labels and the probes to buy. You can buy different chips. There are different chips to do different tests. And you can, after that, these results will be analyzed by computer analysis, and they will give us comparing the patient and the friend, and will give us the results. So by looking at the difference, we'll tell you whether you have variance. The variant can be, in this case, at the numerical level or at the structural level. So like this one, you can detect copy number variants. They have extra copy number variants, extra big number. So now let's look at the second method. The second method here is look at, is there any test that examine DNA or one or more genes? So this is now look at the method that detect DNA or one or more genes. So this is known as a molecular genetic testing. Now this testing for DNA variants, look at DNA variants. The DNA variants can be in the coding or non-coding sequences or in the regulatory elements associated with traits or a genetic disorder. What does that mean? So this is a DNA. So assuming this is a double-stranded DNA. So as I told you, the gene. So this part here has the genetic information for a gene. So this is called a coding sequence. This is for another coding sequence, can be used for the same protein or different protein, different parts of it. Now this green part is called a non-coding. It doesn't code for protein, but it contains DNA sequences, still very important. And some of the sequences are important for regulatory. Regulatory means they control gene expression for gene expression purpose. So non-coding here, although it doesn't code, this region here is also very important. So if there are changes here, if there are changes in DNA sequences, it affects the outcome. Let me explain. So let's say this one DNA, there's one DNA here, and you compare DNA, there's one strand, another strand of another DNA. You compare, you can see that there's one change. A has changed to C. So there is a variant. Either C changes to A or A changes to C. So we have two variants, they are called the variants. So at a specific location, you have nucleotide or base variants called DNA gene variants. So it can be changed. It can be A to C or T or C or G. So So this is referred to as an A allele. This is called a C allele. In this case, the allele. So if you look at this spot here, in a somatic cell, so because you have two together, there are two of them together in a somatic cell, a pair. So this is called the AC alleles. You have alleles A and C. That's how we call it. And this is called the genotype. The genetic makeup, if that says in your DNA, inside your cell, is called you have a genotype of AC. So this is also the change is called a single nucleotide polymorphism, because polymorphism means different form. In order to call it an SMP and not a mutant or a variant, this must be inside a population, at least more than 1% of a population. So you have to look at it. If it's less than that, then it's just a variant, a common variant or a mutant. So, okay, so we can determine them. The genetic testing is to determine whether it's an A or a C. Okay, why is it important to know? For example, there you have, in this case, let's say you have a DNA strand. The DNA strand has T, and another DNA strand has C. Very similar, except for T and C. So you can call a T and C mutation. So this change, does this change affect a gene? What they call, does it cause a change in the trait and may cause genetic disorder? So let's look at this particular DNA. This particular DNA, after transcription in the RNA transcription, you can see C will pair with A, G, A to U. Now T will pair with A. In the RNA, it has an A. And these three letters here, G, A, U, when during translation, it codes for one particular amino acid. So in this protein, it has this amino acid. On the other hand, this strand here has a C. After transcription, C will pair with G, C will pair with G, A will pair with U. So it becomes GGU. It has changed from GAU to GGU. And GGU during translation is used to make a different amino acid, different from this one. So you have now two different polypeptides, two different proteins. One of them may not be functioning, may not be working. So then there is big problem. So that's why one nucleotide change here or one base change can lead to a serious change in the protein, and it may cause a disorder. So, and that's why this genetic screening, genetic testing, genetic profiling, DNA test, SNP or variant genotyping, all these terms actually means the same thing. They are used by different people to mean the same thing, is to identify and analyze DNA variants. We want to know what kind of variants at the DNA level, okay? So this detection can be used for diagnosis to determine the disease in a medical or non-diagnostic, okay? It can be, unlike chromosomal. Chromosomal is always for diagnostic. So for now that we know the molecular variants, how do we determine them? For in the genetic testing here, we have three major testing methods. You can use PCR. You are all familiar to detect, for example, the COVID-19 to determine the virus. You use PCR or RT-PCR, reverse transcriptase. So you can use PCR or real-time PCR or other method. There are many different types of PCR. And normally PCR to detect known single or multiple DNA variants. You can use multiplex PCR to detect many at one time, or you can determine simple copy number variants. These are for simple detection. Now there are more powerful methods. So we can use DNA sequencing technology. In other words, this technology can determine the sequence to determine that every nucleotide in a stretch of DNA, one particular chromosome or the entire genome. So we have different methods of sequencing you have Sanger dideoxy sequencing. That's the first one. And then subsequently, now we have the next generation sequencing, or what you call this, the NGS. And now we have the new one. Some people call it the third generation sequencing, the single molecule long read sequencing. You can determine single DNA and determine very long, or you can determine DNA from a single cell, one individual cell, rather than a mixture of cells, single cell. So these are the newer and better and even cheaper method of sequencing. And the sequencing technology allows us to look at everything. We can look from very simple, a single variant, to a single gene, the single gene sequence, or we can look at a set of genes, or called a gene panel, or we can look at the entire exome. Exome means, the complete coding sequences, all the coding sequences from a genome. That is the exome. We don't look at the non-coding, all the things. So you can entire them. Or we can look at the entire genome. The entire genome means everything. Coding, non-coding, regulatory sequences, we can look through them. So you can use sequencing to look at all these, okay? Very powerful. And therefore, DNA sequencing can detect existing variants as well as can detect de novo. De novo means new, newly created variants. Unlike here, this determine known, can determine variant. And in addition to these two, we can use DNA or genotyping microarray, almost similar like the chromosomal microarray. But here we look at the gene microarray. So for DNA sequencing, now we have machine, they have different type of machine. The machine is very powerful. We can do all sorts of sequencing. You can see here, all kind of sequencing. So microarray, you have a slide, you have the slide, on the slide here, you have spots. And the spots here, you have various small stretches of DNA sequences and for hybridization. And where you can pick up through these colorful spots. So, so that can pick up, microarray can be used to detect different types of single nucleotide polymorphism. So you can buy different types of chips. You can buy the SMP arrays or other type of chips here. So this is one type of chips. And then it depends on your purpose. So you can look at nucleotide variants, for example, individual variants, single genes, or a set of genes, or you can look at the exome. You can look at copy number variants also. Okay, so this is how we do that. I will not want to go into detail because each one that is a lot of detail. We need separate lectures. For you right now, it's important just to know the names. You know what they are. Okay, you can read up if you are interested in. And the last test here for the proteins, for biochemical functional tests. And this is also part of a genetic test. It's not DNA test, but genetic test. These two are the DNA chromosome, and these are the DNA tests and genetic tests. The idea here is to determine the amount of metabolites. As we know, in the metabolism, we have metabolites, and also determine various types of proteins, the level. It can be enzymes, can be also antibodies, for example, or hormones. You can look at abnormalities in them. If you can detect abnormalities, it means it indicates there are changes in to the DNA, and therefore that's resulting in genetic disorder. So it's an indirect way to indicate damage in DNA. So, biochemical DNA tests, you're very familiar. When you go for your annual checkup, body checkup, most of the time the doctors will take your urine, will take your blood to do tests, biochemical tests. So they want to test your body function. So these are the diagnostic clinical biochemistry. So this is when a doctor collects your urine or your blood to do the test, as part of this diagnostic test. So this is using the biochemical knowledge and techniques to assist in the diagnosis of human disease. Okay, so when you do the test, when you collect your blood and urine to do the test, you are actually doing assay. assay for and to evaluate what? Absence or presence or functions of some important metabolic enzymes, vital for health. Or you look at some important metabolites, for example, ammonia and other metabolites, okay? And even you look at it for the blood, all right? Functional capacity of critical organs and systems such as liver. We know certain organs like liver, heart, kidneys, certain enzyme must be there, certain metabolites must be there. You can detect them. For example, here, this one, the very common biochemical test will be the newborn baby. Every newborn baby will be automatically tested for all these biochemical tests. See, the newborn screening test, they are biochems. You can see that the doctor will make a prick at the heel to collect drop of blood. And then the blood will be used to do this test to look at the amino acid disorder, phenylketonuria, maple syrup, urine disease. It smells very nice. The urine, maple syrup, but it's a disorder. Fatty acid oxidation disorder, organic disorder, hemoglobin, blood disorder, sickle cell, thalassemia, endocrine, all the hormones and miscellaneous. So you can see that all these are very done automatically. Even in Malaysia, every newborn babies will go for all these tests because this will tell us the gene, the status of the gene. So why do we need, I'll finish with you now, the thing now, why do you need to know all these tests? In our lifetime, before we are born in fetus, until we are adults, at different stages, there are the principles of genetics and genome have been applied to medical care and personal health at different stages. It can be used for diagnosis, management, treatment of hereditary and genetic disorders, metabolic disorders, and then also for counseling, genetic counseling of people about if you have genetic disorders, they will need counseling. Okay, so all the tests that I mentioned earlier on, they can be used to predict disease and health disease. So now in terms of tests, you can order, there are a range, a plethora of genetic tests for better diagnosis and clinical management. So for example, carrier testing. If you know you have certain disease runs in your family, before, if you are married, before you decide to have a baby, you may go for carrier testing. So this will be at the adult stage, before you have to have pregnancy. You do a carrier test, whether you carry certain bad genes or not in husband and wife, or prenatal testing. After when a baby is in the womb, you can do tests prenatal before a child is born. What kind of, whether you're normal or with certain diseases, genes. Pre-implantation, if you're doing in vitro fertilization, test your babies. So after fertilization in the embryo, you want to do the test. You want to know whether the embryo carries any serious genetic disorders or not before you decide to plant into the egg of the mother. Okay, newborn screening. I told you just now, after birth, the baby will be screened, and then subsequently, when they are young here, you do predictive testing. Especially if your family runs you have certain family diseases, you do predispositional. They will be predictive. You have to say whether it's going to happen or not. Predispositional means may or may not happen. Pre-symptomatic, it will definitely happen. If you have certain diseases, then Huntington disease that happens to somebody, then you know, most likely if the gene is there, it will happen to a person. So you can do transplantation testing to check whether your genes, diagnostic, for forensic, for pharmacogenomics. So all these can be done, but this must be done through healthcare, through doctors. You cannot order on your own. Okay, this one. Another type now, the last type here I want to talk about is because now with the era of digital consumerism, you can order online, direct order of testing. So there are companies that market various type of direct to consumer, personalized, non-diagnostic. It's not for diagnostic, non-diagnostic genetic test for healthy person. You don't have to be sick. But you can order to check for your ancestry, for your kinship. You can look for high style genes which are related to lifestyle and trait. And then for nutrigenomics, anything your nutrition, diet. So personalized nutrition. All this you can order. Okay. And so the last topic here is you can, before you go for all these things, you should also be aware. So you must be familiar. I've given you the process, so at least now you're familiar with the process before a genetic test is done. So, but what very important, if you want to do a genetic test, if you know your family has a certain genetic disease, then they will need to gather medical family, a family medical history. And this may be stressful because they will talk to family members. Some family members don't want to discuss about the diseases. Okay. And you must know that genetic data are personal. They are very private, confidential. And the other thing is there is always a respect for privacy, for autonomy, for personal best interest, for responsibility, for the health of the future. You do a carrier test, for example, for the future generation. This is what I call the reproductive choice or liberty of individual based on the information that you have obtained. And also you must have respect for genomic difference. People are different. And also remember, if you do a genetic test, your data are of interest and relevance to family members and relatives. Your parents may want to know, your sister, brothers want to know, your cousins may want to know, the uncles may want to know. For example, you test for cancer, a breast cancer gene if you are female. So whether you want to share of them or not, because some of them they want to know, but then they may have negative impact on your family and may have negative impact on personal relationship. This sort of knowledge, some people really don't want to know. After knowing they have actually all kind of things. So be aware of the risk. You also must, from there you know the risk associated with genetic testing. And once you know the risk, there is emotional, social, financial consequences regarding the test. Okay. And then finally here, the results sometimes the result may return inconclusive and uncertain, then give you stress. So, and make sure you have emotional support. There are people to give you support. And you must be able to handle stress, anxiety, and potential stigmatization, because certain diseases may have certain stigma when people know about it. If they don't know, that is fine. So you must learn how to deal with medical doctors or clinical geneticists. Look at this one, or genetic counselor. So these are the people you have to be very frank with them. You have to discuss with them. They will know your result. They will advise you. These are the healthcare professionals. They are very professional. So they keep your secret. They won't discuss other people. But more important is, do your employers, insurance company need to know, or do they have the right to know? So all these things, they are what they call the ethical, legal, social implications. So all these things. So it's very important you should know all this before you go for any testing, okay? And okay, that's my talk. Thank you very much.
Thank you very much. Thank you for Dr. Cole. Let us learn about gene testing, how important for our life. Okay, thank you very much.